Hello student, I am Dr. Amar Vanzari and I am assistant professor in zoology at Moge College, Pandrakoda. Student, in this video I am going to demonstrate the practical syllabus. In B.Sc. second year same third, you have to study the paper having the name Life and Diversity and Animal Evolution. मतलब आपको इस साल दो पार्ट स्टडी करने हैं. That is one chordate animals and second one animal evolution. In this practical syllabus, there is a 5 parts. In part 1, there is taxonomy. Taxonomy means study of naming, their classification and their descriptions. मतलब इस chordate animals में जितने भी animals है उनकी आपको classification करनी है, उनका आपको description करना है. In part 2 there is a dissection. In dissection we have to do anatomical study. Anatomical study में हम different organ system की study करते हैं. Student 2014 तक dissection यह part हमारे syllabus में था, लेकिन सेंट्रल गवर्नमेंट ने 2014 के बाद इस dissection पे बैन लगा दिया. Nowadays we do the demonstrations with the help of charts, models and computers. In this part, we have to study the internal organ system of Scoliodon, rat and air bladders in fishes, and also there is a part of mounting. In part 3, there is a study of osteology. Osteology means study of bones, or it is also called as the endoskeleton. So, in this part, we have to study the osteology of rabbit and varanus. In part 4, there is a study of animal evolutions, how our animals are evolved. So in this part, there is a study of fossils, there is a study of evidences of evolutions, and also there is a study of Mesozoic Era and also mimicry and colorations. And in last part, there is a study of beak and legs modifications with reference to birds. In part 5, there is histological study. In histological study, we do the microscopic study of cells and tissues of different organs. So in this histological part, we have to study the histological parts of Amphioxus, then frog and rat. So let's start to study practical syllabus in details. As we know that on our earth there is life, means there is 70 million living species on our earth. Out of them 7 million species belongs to kingdom Animalia. 7 millions मतलब 70 lakh. These 70 lakhs animals are divided into two groups, that is non-chordate and chordate. 98% animals belongs to group non-chordates, while only 2% means near about 60 to 70,000 animals are chordate animals. All these chordate animals shows variations in their body form or body structure. This variations is called as diversity of chordate animals. These chordate animals, as we know that, they have a presence of notochord. Notochord is a rod-like skeleton which is used to support the body. अभी हमारे बॉडी में नोटोकॉर्ड absent है, but it was present in embryonic period. जब हम माँ के पेट में थे तब वहाँ हमारे बॉडी में नोटोकॉर्ड था, लेकिन जैसे ही हमारी ग्रोथ हुई दैट नोटोकॉर्ड गेट कन्वर्टेड इनटू वर्टिबल कॉलम्स. सेकंड मेन कैरेक्टरिस्टिक्स इज प्रेजेंस ऑफ फैरेंजियल गिल स्लिट्स. आप लोगों ने फिशेस में ऑन आइदर साइड ऑफ फैरेंक्स यू कैन सी द ओपनिंग्स ऑफ गिल्स, दैट इज कॉल्ड एज द गिल स्लिट्स. अभी हमारे बॉडी में गिल स्लिट्स नहीं है, लेकिन जब हम एम्ब्रियोनिक स्टेज में थे तब हमारे भी बॉडी में गिल स्लिट्स प्रेजेंट थे, एंड these gill slits then disappears. Then third characteristics is the presence of single dorsal tubular nerve cord. Nerve cord is a bundle of neurons which get converted into brain and spinal cord. And the fourth main characteristics is the presence of heart on ventral sides. The phylum Chordata is divided into two groups, lower chordates or protochordates and second one higher chordates or craniata. Protochordate animals are the first evolved chordates animals which are evolved from non-chordate animals जो पूरी तरह से evolved नहीं हुए लेकिन उनमें chordate के characters पाए जाते हैं और इनमें हमें हेड पार्ट्स नहीं दिखाई देता है क्योंकि इनमें क्रैनियम कैविटी इज अब्सेंट, इसीलिए इन्हें अक्रैनिएटा भी कहा जाता है. दिस प्रोटोकॉर्डेट इज डिवाइडेड इनटू सबफाइलम यूरोकॉर्डेटा and cephalochordata. In urochordate animals, the notochord remain present only in the tail region of larva. Larva is free-swimming animals and after metamorphosis, means after growth into adults, the tail loses and head settle down at the bottom of oceans. So, in adult stage, we only found the head regions. These animals are totally marine animals and whole body is covered by a thin layer called tunic. Example is Herdmania and Doliolum. Subphylum Cephalochordata animals are the small fish-like marine animals where notochord remain present throughout the body. In these animals, pharynx is big and gill slits are present. Example is Amphioxus. In Urochordata and Cephalochordata, there is an absence of cranium cavity, means true head remains absent. In these protochordate animals, notochord remain present थ्रूआउट दी लाइफ, मतलब नोटोकॉर्ड का कन्वर्शन वर्टिबल कॉलम में नहीं होता है, इसीलिए इनको वर्टिब्रेट्स नहीं कहा जाता है. द नेक्स्ट ग्रुप इज हायर कॉर्डेट्स और क्रैनिएटा which is also called as a subphylum vertebrata. The vertebrata is divided into two divisions, agnatha and gnatostomata. A means absent and gnatha means jaws, means these animals are without jaws. This agnatha is divided into two classes, ostracodermy and cyclostomata. In class cyclostomata, mouth is without jaws and they can't bite. So they are provided with suckers and they are also called as ectoparasites, which sucks the blood of host animals, that is the big size fishes. Their skins are soft and without any scales. Endoskeleton is cartilaginous and they have an absence of paired appendages, means there is absence of fins or foot. Example is Petromyzon and Maxine. Gnatostomata animals are having the jaws. These are also divided into superclass Pisces and superclass Tetrapoda. Superclass Pisces is divided into two classes. One is the Chondrioches, means they have endoskeleton made up of cartilage, and second one is the Osteoches, means they have an endoskeleton made up of bones. These fishes are totally aquatic animals, means they found in marine as well as fresh waters. They have a spindle-shaped body to minimize the water resistance. They are also cold-blooded, so it is also called as a poikilotherms animals, means they can change their body temperature according to surrounding environments. They have a paired and unpaired fins for the purpose of swimming. Tail fins is useful as a steering organ. Exoskeleton is present, which is made up of scales, and endoskeleton, it may be cartilaginous or bony. Example is Scolodon, seahorse, electric ray, labio, and stingray. Superclass Tetrapoda is divided into four classes. Class 1 is the Amphibia. Amphi means both and bia means life. Means they have aquatic as well as terrestrial life. Their larval life is aquatic, that is also called as a tadpole, which look like the fish, and respiration is aquatic through the gills. In adult life, these gills disappears and they develop the lungs. In water, they respire by thin skins and also by buccopharyngeal cavity. And in terrestrial life, they respire by lungs. Also, they have two pairs of appendages. Exoskeleton is absent because there is absence of scales or hairs, and their skins remain moist because of the presence of mucus gland. Example is frog. फ्रॉग जिनकी स्किन स्मूथ होती है, देन टोड जिनकी स्किन रफ होती है, सैलेमेंडर एंड इथियोफिस. Second class is the reptiles. They are evolved from the terrestrial life. So they are the first true terrestrial animals with creeping movements, and they can't lift their body upwards. Skin is dry and scaly. Neck is present between head and trunk. Internal ear is absent and digits are provided with claws. Example is tortoise, turtle, snakes and lizard. Class Aves are well adapted to aerial lives. Their forelimbs are modified into wings. They are warm-blooded animals, body tapering at both ends to resist the air while flying. Exoskeleton is feathers. Anterior part of mouth is divided into beak. Example is peacock, pigeon, duck and penguin. Class Mammalia is well developed for all habitats. Their special important character is presence of mammary glands in female, presence of external pinna and presence of hairs. They are also warm-blooded animals, that is the homeothermic animals. Body is divided into head, neck, trunk and in some animals tail. Exoskeleton is hairs or fur. Example is whale, bat and man. In part 2, for anatomical study, there is a dissection or demonstration. For the dissection, we use the tools, that is the scissors, forceps, probes, needles and scalpel. In this part, there is a dissection of afferent and efferent blood vessels of Scolodon. As we know that blood vascular system includes heart, blood vessels and blood. There is two types of system to circulate the blood in the body, that is the arterial system and venous systems. Arterial system is used to carry oxygenated blood away from heart, and venous system it is used to carry deoxygenated blood from different body parts towards heart. In Scolodon, there is respiration by gills or branchial respirations So arterial system, it is divided into two categories, that is the afferent branchial artery and efferent branchial artery. Afferent branchial artery arises from heart, which brings deoxygenated blood to gills for oxygenation, and efferent branchial arteries originates from gills and gives oxygenated blood to different parts of body. Second dissection or demonstration is of cranial nerves. Nervous system of Scolodon is divided into three parts, that is the central nervous system, which includes brain and spinal cord. Second one, peripheral nervous system, which includes cranial nerves and spinal nerves. And third one is the automatic nervous system. So in this demonstration you have to demonstrate cranial nerves, which is 10 pairs, and it goes into different body parts. Next demonstration is of internal ear of Scolodon. As in fishes, there is absence of external and middle ear. So this internal ear is used to change the sound waves to electrical signals, and it allows brain to hear the sounds. It is also important for the balance. It consists of three semicircular canals. One is the anterior vertical semicircular canal, second is the posterior vertical semicircular canal, and third one is the horizontal semicircular canal. Next anatomical study is of rat, which has objective to compare rat anatomy with human anatomy. Here we have to study the digestive system, reproductive system, venous system, and arterial systems. In this organ system of rat, there is same organ which are present in the organ system of man. Next demonstration is the observation of air bladders in air breathing fishes. These air breathing fishes is also called as the amphibious fishes. They are able to respire in air. Many fishes living in oxygen poor water develops air breathing lungs. Next part is the mounting or permanent micropreparations of animal cells or tissue. Mounting, the cells or tissues remain as such without undergoing major changes in their structure or functions. For the mounting, there is a procedure, that is, first is the killing or narcotizations of animals. Then we dissect out the tissues, and then it fix into fixations, then washing that tissues, then staining by using the different stains, then destaining, and then dehydrations means removal of water, and last we have to mount that tissues on the slides, and last one is the labeling. So in this year you have to prepare a permanent micropreparation of fish scales, that is the placoid scale, ctenoid scales or cycloid scales, and also there is a mounting of ampullae and eyeball muscles. In part 3 there is osteology of rabbit and varanus. Osteology means study of bones or endoskeleton. All the skeleton of body is divided into two parts. One is axial skeleton and second is appendicular skeleton. Axial skeleton forms the central axis of the body, and it consists of skull, vertebral column, ribs and sternum. While appendicular skeleton is added to axial skeleton for the purpose of locomotion, and it consists of anterior limbs which are attached to the pectoral girdle means shoulder, and lower posterior limbs are attached to pelvic girdle means hip. For the convenience of body, four arrangement has been followed. Number 1 is the skull, vertebral column which consists of different vertebrae. Third one is the girdle, that is the pectoral and pelvic girdle, and fourth one is the limb bone, that is the forelimbs and hind limbs. In part 4, there is a study of animal evolution. Evolution is the slow and gradual changes in a species from long times. Means for evolution it takes millions of years. In this part, there is a study of fossils. Fossil is a plant or animal from long ago which has turned into stone, while living fossil is a living species of organism that appears to be similar to fossil. Example of living fossil is lemurus. There are different types of fossil. Number one is trace fossil. This fossil shows evidences of activity. Example is footprints. Second one is the mold fossils. They show an impression of the animal or plant on the stone. Third is the cast fossil. This shows an image of animal or plant. Next is two form fossils, means there is actual plant or animal is the fossils, means mummy. Next is petrified or pre-mineralized fossils. In this, there is empty pore spaces are filled in by minerals such as in petrified woods. Next, you have to study evidences of evolution. These evidences can be structural or molecular level, means anatomical or biochemical level. In evidences of evolution, you have to first study the connecting link. Animals exhibiting characters of two adjacent taxonomic groups are called connecting links. Example is Archaeopteryx, which is the connecting link of reptiles and birds. Second one is the Lemurus, which is the connecting link of Annelida and Arthropoda. There is evidences of evolution by comparative anatomy. In this, you have to study homologous and analogous anatomy. In animals, homologous structures are that structure that are comparatively similar to each other but have different functions, and they have similar genes descend from a common ancestor. Example is the limbs of vertebrates, means turtles, alligators, birds and mammals. Means the limbs of these animals are similar but it may be used for different purposes. Analogous structure are that structure that looks similar but have different origins. Example is the wings of bat, birds and insect. There is some embryological evidences, that is embryos of different species may appear similar in early stages of development. That is, the development of embryos of all vertebrate animals are similar, and second, there is relationships to a common ancestor. Means all vertebrate animals are developed or evolved from a common ancestor. In next part, there is study of adaptations of Hardy-Weinberg equilibrium law. In the evolution of species, population of that species play important role. So, study of population genetics is essential for understanding the species adaptations and evolution. For better understanding of population genetics, Hardy-Weinberg law should know. This law states that the allele and genotype frequencies in a population remain constant
generation after generation if there is no selection, no mutation, no migration or random drift in a populations. It describes populations that are not evolving and does not show evolution. This law explained in an equation. Then there is study of Mesozoic reptiles with the help of models and charts. The Mesozoic era is the middle life of Earth, which begins from 252.2 million years ago, and this era is also called as a age of reptiles because in this era dinosaurs ruled the earth and dinosaurs dominated the earth. When mass extinction occurred, it killed off all the dinosaurs are killed, and this was the largest in the history of earth. There was many groups of reptiles which contained many species, that was dinosaurs, marine reptiles, lizards, tortoises and mammals was not much evolved. In next there is a study of mimicry and colorations in animals. Mimicry means similarities between the species. There is different types of mimicry. One is protective mimicry, there is cancelling mimicry and warning mimicry. In some animals there is aggressive mimicry. In these predators or parasites species resembles non-threatening species. In Batesian mimicry, palatable animals mimic the bad tasting. Mullerian mimicry is done when two or more poisonous organisms resembles each other. Atomimicry occurs in intraspecific organisms, which occurs when a single species mimics the part of its own body. Then you have to study colorations. One of the morphological character of an animal is colorations. Animals living in tropics shows wide range of colors. Coloration may be due to physical colors, that is the refractions of light rays on the body surface, or it may due to chemical colorations, which is due to presence of color pigments. In next part you have to study beak and leg modification in birds. The shape of beak of birds are evolved or modified on the basis of what they eat. Means some birds eat seeds, fruits, insects or nectars or the fishes or some mammals. So they are modified their beaks according to food habits. Also the feet or legs of birds are evolved or modified according to habitat where they get food and they do important activity. That is, some birds lives in water, so their legs are modified for the purpose of swimming. Some birds develop their legs for the catching prey. Some are developed for walking. Some legs are developed for perching, wading or climbing. This beak and legs modification in birds allows them to survive in that habitat. In fifth part there is histological study. In this histological study you have to study the microscopic cells or tissues of three animals, that is the Amphioxus, frog and rat. You have to study TS of liver, TS of stomach or TS of intestine. For histological study of reproductive organ, there is study of TS of ovary and TS of testes. For the histological study of endocrine system, you have to study the TS of pituitary gland, TS of thyroid gland or TS of adrenal gland. And also there is study of TS of kidneys, TS of lungs. Hope you like this video of demonstration. If you like it, please share to your friends and also comments and subscribe the channel. Thank you.